Hello everyone. Today we will discuss about lipoproteins. Lipoproteins are compound lipids that are formed by the combination of lipids with proteins. So, a lipoprotein is a biochemical assembly of lipids and proteins. Here, the protein part is known as apolipoprotein or apoprotein. And the lipid part is the prosthetic group. This is the prosthetic group. This lipoprotein helps to transport the hydrophobic lipids to various tissues. This is for utilization and storage. That is, the lipoproteins serve as a carrier of lipids in plasma. So, let us see the structure of lipoproteins. Lipoproteins have an inner core of non-polar lipids, which usually contain triacylglycerol and cholesterol esters. So, lipoproteins have an inner core. Inner core of non-polar lipids. This contains triacylglycerol and cholesterol esters. These are cholesterol esters. Here it is triacylglycerol. This inner core is surrounded by a monolayer of amphipathic lipids. Inner core is surrounded by a monolayer of amphipathic lipids. The amphipathic lipids mainly phospholipids and cholesterol. The inner core is surrounded by a monolayer of amphipathic lipids. This is the monolayer of amphipathic lipids, and this contains mainly phospholipids and free cholesterol. In the inner core there is cholesterol ester, and surrounded by a monolayer it contains phospholipids and free cholesterol. This is phospholipid and this one is free cholesterol. These phospholipids, the polar head is on the outer side. And the non-polar tail on the inner side. Polar head on outer side and non-polar tail is on inner side. Then in the outermost layer there is peripheral apoprotein. In the outermost layer there is a peripheral apoprotein. This is peripheral apoprotein. For example, ApoC. Then there is an integral apoprotein. Here there is an integral apoprotein. Integral apoprotein. For example, ApoB. This is the entire structure of these lipoproteins. It contains an inner core of non-polar lipids which contain triacylglycerol and cholesterol esters. This inner core is surrounded by a monolayer of amphipathic lipids, mainly phospholipids and free cholesterol. The phospholipids the outer side is the polar head and non-polar tail on the inner side. Then the outermost layer there is a peripheral protein, the peripheral apoprotein, that is ApoC, for example ApoC. Then there is integral apoprotein, for example ApoB. This is the structure of lipoproteins. Coming to the classification of lipoproteins, lipoproteins can be classified based on density, electrophoretic mobility and apolipoprotein content. Classification is based on density, then electrophoretic mobility, and on apolipoprotein content. So first we will see classification based on density. So as the proportion of lipid to protein in lipoprotein complex increases, density decreases. Proportion of lipid to protein increases, density decreases. Using this, lipoproteins can be separated by ultracentrifugation. Ultracentrifugation. So lipoprotein with the highest density will settle down at the bottom. Using ultracentrifugation. So in the bottom it will be free fatty acid complexed with the albumin. So this free fatty acid with albumin, usually these are not included in lipoprotein classification because they are loosely bound. So if this is a test tube, here it will be free fatty acid complexed with the albumin. Free fatty acid complexed with albumin. Usually these are not included in lipoprotein classification because they are loosely bound. Then above that will be HDL. This is HDL. Then after that LDL. Then it will be IDL. Then it is VLDL. And here it is chylomicrons. It is chylomicrons. So this is the classification based on density. So lipoprotein with the highest density will settle down at the bottom. So in the bottom it will be free fatty acid albumin. After that HDL, LDL, IDL, VLDL and chylomicrons. So coming to the density, chylomicrons having density less than 0.95 gram per liter, density in gram per liter. Then HDL, VLDL, chylomicrons 0.95, VLDL is having density 0.95 to 1.006, 0.95 to 1.006. Then next one is IDL having density 1.006 to 1.019. Then LDL, it is having density 1.019 to 1.063. Then HDL having density 1.063 to 1.215. And this free fatty acid complex with the albumin, it is having density greater than 1.215. This is the density of individual lipoproteins. So this is the classification based on density. Then second classification is based on electrophoretic mobility. So based on electrophoretic mobility, this is based on the size and protein content. Electrophoretic mobility. This is based on size and protein content. Size and protein content. So higher the protein content, higher will be the mobility. Higher the protein content, higher will be the mobility. This chylomicrons have least protein content. These have least protein content, so it will remain in the origin. It will remain in origin. Then according to size, chylomicrons are bigger in size. Chylomicrons are bigger in size, and HDL is smaller in size. So let's see the classification based on electrophoretic mobility. This is positive, negative. So the chylomicrons have least protein content, so it will remain in the origin. So if this is the origin, chylomicrons will be here. In the origin. Then next will be LDL. Here it is in the positive side, that is free fatty acid with albumin. Free fatty acid complexed with the albumin. This is free fatty acid albumin. Then next will come HDL. Next is HDL. HDL or alpha lipoprotein. HDL is known as alpha lipoprotein. Then VLDL will come. This is VLDL, and it is pre-beta lipoprotein. Pre-beta lipoprotein. Then IDL. Next one is IDL. This is known as broad beta. Broad beta lipoprotein. Then here comes LDL. This is LDL. It is known as beta lipoprotein. Beta lipoprotein. Here in the origin it will be chylomicrons. This is chylomicrons in the origin. So based on electrophoretic mobility, this is based on its size and protein content. Higher the protein content, higher will be the mobility. Chylomicrons have least protein content, so it will be in the origin. Then chylomicron, according to size, chylomicrons are bigger in size. HDL is smaller in size. So based on electrophoretic mobility, here it is free fatty acid albumin. Then next will come HDL will come. Then HDL, after that VLDL. HDL is alpha lipoprotein, VLDL is pre-beta lipoprotein. Then comes IDL, that is intermediate density lipoprotein, that is broad beta. Then LDL, that is beta lipoprotein. Then chylomicrons. HDL is high density lipoprotein. Then VLDL, it is very low density lipoprotein. IDL is intermediate density lipoprotein. LDL is low density lipoprotein. Then next classification is based on apolipoprotein. Apolipoprotein is the protein part. So based on the apolipoproteins, based on apolipoproteins, there are 5 types. First one is lipoprotein A. Here the apolipoproteins are A1 and 2, A1 and A2. Then next one is lipoprotein B. Here the apolipoproteins are Apo B48 and Apo B100. Next one is lipoprotein C. Here the apolipoproteins are Apo C1, C2 and C3. Then lipoprotein D, it is Apo D. Next one is lipoprotein E. This is Apo E. This Apo E is arginine rich. This is arginine rich. So this is the classification of lipoproteins. So next we will see characteristics of individual lipoproteins. Characteristics. So first one, chylomicrons, VLDL, then comes IDL, LDL and HDL. If we consider size, chylomicrons are bigger in size. Chylomicrons are bigger and HDL is smaller in size. Then density, chylomicrons are less dense. In the HDL, it is highly dense. Highly dense. Then source is chylomicrons, these are synthesized in intestine. VLDL in liver. Intermediate density lipoprotein, this is formed from the degradation of VLDL and HDL. Degradation of VLDL and HDL. Then low density lipoprotein, these are synthesized from VLDL. Then HDL, these are synthesized in liver and intestine. Liver and intestine. These are the sources of these individual lipoproteins. Then next characteristics, major apolipoprotein present. Major apolipoprotein. In chylomicrons, Apo A, Apo B48, Apo C and E. These are the major apolipoproteins present in chylomicrons. Apo A, Apo B48, Apo C and E. In this VLDL, it is B100, C123, C1, C2 and C3. In the case of IDL, it is B100 and Apo E. B100 and Apo E. In the case of LDL, it is B100. B100. Then HDL, it is Apo A, C, D and E. Apo A, C, D, E. These are the major apolipoproteins present. Next one is protein content. Protein content in chylomicrons, it is 0.5 to 2%. In VLDL, it is 7 to 10%. In IDL, it is 11%. In LDL it is 25% and in HDL it is 50%. It is 50%. That is a protein content. 0.5 to 2%, 7 to 10%, 11%, then 25% and 50%. Then lipid content in chylomicrons is 98 to 99%. 98 to 99%. Here it is 92 to 93%. Here it is 89. In IDL it is 89%. In LDL it is 75%. And here it is, in HDL it will be 50%. That is the lipid content in lipoproteins. Then major lipids in chylomicrons are triacylglycerol, then phospholipid and cholesterol. These major lipids in chylomicrons are phospholipid, triacylglycerol and cholesterol. Then in VLDL, in VLDL it will be triglyceride. In IDL it is cholesterol. Then in LDL also cholesterol, and in HDL it will be phospholipids and cholesterol. Phospholipids and cholesterol are the major lipids present. Then coming to the function, chylomicrons usually transport exogenous triglyceride. It transports exogenous triglyceride from GI tract to muscle and adipose tissue. From GI tract to muscle and adipose tissue, that is peripheral tissues. That is the function of chylomicrons. Then coming to VLDL, main function is to transport endogenous triglyceride. Transport endogenous triglyceride from liver to peripheral tissues. From liver to peripheral tissues, that is muscle and adipose tissue. Then this IDL, its function is similar to LDL, and it transports triglyceride and cholesterol. That is a function of IDL. Then coming to LDL, its function is this LDL is usually the metabolic end product of VLDL. Its function is to transport cholesterol from liver to peripheral tissues. Cholesterol from liver to peripheral tissues. That is the function of LDL. Then coming to HDL, that is important, that transport cholesterol from peripheral tissues back to liver. Cholesterol from peripheral tissues back to liver for degradation. So this is the these are the characteristics of lipoproteins. Then another function of these lipoproteins are they are enzyme cofactors. They are enzyme cofactors. Enzyme cofactors and can act as activator or inhibitor of the enzyme. They can act as activator or inhibitor of the enzyme. For example, activators ApoA1 activates LCAT, that is lecithin cholesterol acyltransferase enzyme. Then ApoC2, it activates lipoprotein lipase. Lipoprotein lipase. Then inhibitor ApoA2 and ApoC3. ApoA2 and ApoC3. This inhibits lipoprotein lipase. Lipoprotein lipase. So these are act as enzyme cofactors. Then ApoD, it is an important factor in human neurodegenerative disorders. ApoD is an important factor in human neurodegenerative disorders. So these are the major characteristics of lipoproteins. So this is all about lipoproteins. This is today's topic. Thank you for watching.